What we'll do here is take a look at microbial genetics. One of the ways of defining genetics is that it's the study of a cell's DNA and everything that that DNA can do for a cell. So that's what we'll do here at the beginning. We'll just basically review, what does DNA do? And so what you're taking a look at here is a bacterial cell. In that bacterial cell, you see some of its ribosomes, and also inside you see its DNA. So remember that bacteria, like all prokaryotic cells, have circular DNA. DNA is a type of nucleic acid, which means that it's a string of nucleotides that have all been covalently bonded to one another. Remember that that long string of nucleotides can be divided into smaller strings or smaller packets of information called genes. And what each one of those genes does is that it contains the information that's needed in order to tell the cell how to make one particular type of protein. Remember, the spots in the cell where the proteins are actually produced are the ribosomes. So somehow the information has to be able to get from the DNA over to the ribosomes, and once that information is there, it's then used to construct different types of proteins. The reason why proteins are important to a cell is because, well, proteins can do all kinds of different things, right? So there's some proteins that are involved in helping to maintain the structure of the cell. There's some proteins that act as enzymes. There's some that act as pores. There's some that are involved in motility. So proteins do all kinds of different things that are needed in order to keep the cell alive. So basically, DNA is important because it tells the cell how to make proteins. In order for that protein to be made, somehow the information has to be able to get from here over to here. And so if you remember from cell biology, the way that that happens is by way of a type of molecule called messenger RNA. So if a cell is going to make a protein, the first thing that happens is that the information on that DNA is used to make something called the messenger RNA. And basically, I see that messenger RNA as a cheap copy of the DNA. They both do the same thing, except that messenger RNA can go over to the ribosome. And once it's there, the information that's found within that messenger RNA then is used to tell the cell how to make the corresponding protein. So what we're saying here is that making a protein, it's a two-step process. During the first step, that DNA is used to make a messenger RNA. That step is called transcription. And it makes sense that this process is called transcription because the root word for transcription is transcribe. And transcribe means to basically make a copy of something. And so that's what's happening here. You have DNA, which is a type of nucleic acid, that's being used to make another type of nucleic acid, RNA. Right? So these are basically two of the same thing. They have the same information stored within it. This second step of making a protein is called translation. So the step where the messenger RNA is then actually used at the ribosome to make a protein. That's called translation. And it makes sense that this process is called translation because when you translate a language, for example, you're going from one language to a completely different language. And so that's what's happening here. You have a molecule that's a nucleic acid that's being used to make a completely different molecule or a protein. Right, so DNA is important because it's used to make proteins. Making that protein is actually a two-step process. It involves transcription and then translation. Here's another thing that can happen to a cell's DNA. So there are instances where the cell might decide to make an exact copy of its DNA molecule. And so whenever you have that, DNA is used to make another DNA. That's called DNA replication. And the reason why a cell might be interested in replicating its DNA is so that when that cell divides, the two daughter cells that end up being produced have their own copy of that genetic information that it can use to make all of the necessary proteins in order to stay alive. So to sum up, what does DNA do? Why is it so important? Its primary function is that it instructs the cell how to make messenger RNA. And remember that the messenger RNAs then go over to the ribosomes to make the proteins. And another thing that a cell can do with its DNA is that it can copy it. Right? And so it can be replicated to make an identical DNA molecule. The reason why it makes a copy of its DNA is so that the copy can be passed on to its offspring. So remember to keep these terms separate from one another. So one of the things that I see happening fairly often is that people get those terms transcription, translation, replication mixed up. So someone might say transcription when they really mean replication, or they might say translation when they really mean transcription. So make sure you have a clear idea of what each one of those terms means, because we're going to be using those terms quite a bit when we talk about microbial genetics, and also when we take a look at viruses next week. So here's what we'll do next. We'll focus in on this particular function of DNA, which is to make proteins ultimately. Remember that that process involves two steps, transcription and translation. What we'll do for the rest of this video is focus in on this one step right here. So we'll focus in on what happens during transcription, where DNA is used to make a messenger RNA. So to do that, how about if we redraw the DNA molecule so that you can see its double-stranded structure. So remember, DNA is double-stranded. The two strands are complementary to one another, and that's what allows them to hydrogen bond with one another to form the double-stranded structure. So if a cell is going to read this DNA in order to make a messenger RNA out of it, what has to happen is that the DNA has to become single-stranded. So the two strands have to separate, and that then allows the cell to actually read that information and use it to construct a messenger RNA molecule. The name of the enzyme that does that is called RNA polymerase. And so another thing that I often see people have a hard time figuring out is the difference between the different types of polymerases that you might find inside of a cell. So whenever you see that term polymerase, that tells you that it's an enzyme that makes some sort of nucleic acid. To let you know what type of nucleic acid that it's making, you just take a look at the term in front, right? So if you see an enzyme that's called RNA polymerase, that tells you that it's an enzyme that makes RNA. You might remember that there's also an enzyme called DNA polymerase. So DNA polymerase tells you that it's an enzyme that makes DNA. Right? So they actually make it pretty easy for you, right? That the term in front of polymerase, it tells you exactly what that polymerase is going to make. So the RNA polymerase makes RNA during transcription. This RNA polymerase knows where to first bind the DNA because of a sequence that's found on that DNA called the promoter sequence, or the promoter region. So what that promoter does is that it's a DNA sequence that again tells the RNA polymerase where to first bind in order to begin the process of transcription. Promoter regions are positioned on the DNA so that they're right in front of a gene. And so the gene is the actual sequence then that the RNA polymerase reads in order to make the messenger RNA, which is then used to make the protein ultimately. Right, so the promoters where the RNA polymerase binds so that it can begin the work of making a messenger RNA by using the information on that gene. And so that's what it does. It just travels down the gene and constructs a messenger RNA based off of that information. It does that by inserting the complementary RNA nucleotide opposite each one of these DNA nucleotides. So you might remember that the complement of G is C. So the RNA polymerase will help that C, RNA version of the C, to become attached. When it sees a C, it'll help the RNA version of G get attached. Whenever it sees an A, remember that the complement of A is T, which is a thymine, but RNA contains uracils instead of thymine. So if an RNA polymerase runs across an A, what it'll do is help a uracil attach opposite it. So the RNA polymerase just does that. It goes down that entire DNA strand, reads it, and it helps the complementary RNA nucleotide attach opposite it. It does this until it reaches the end of that gene, and when it does so, the RNA polymerase ends up falling off. It disengages, and the sequence that kind of signifies to that RNA polymerase that you have reached the end of the gene, it's called the terminator sequence, or the terminator region, and here again is what it does in words. After that messenger RNA polymerase has been, after that messenger RNA has been constructed, what happens is then it falls off, it disengages from the DNA. The two DNA molecules can actually come back together so that it can be used to make other messenger RNA molecules. And again, keeping the big picture in mind, the reason why this messenger RNA is important is because it travels to the ribosome, and it's at the ribosome that that information is used to construct a protein. And proteins are just a string of amino acids that have been bonded to one another. Right, so this is, in a nutshell, the process of transcription, where the information on DNA is used to make messenger RNA, and this is basically what happens in transcription in all cells, all eukaryotic cells and all prokaryotic cells. We'll see a bit later that there are some differences, though, in transcription in bacteria versus eukaryotes, and so we'll take a look at that in a bit. But before we do that, what we'll do next is take a look at more of the detail of transcription. And so the first detail we'll look at involves the promoter. So we'll zoom in on the promoter region, take a look at what it looks like, and also what is it about the promoter that helps RNA polymerase attach there. So what we'll do is redraw this portion of the DNA so that we're focusing in mostly on the promoter region. All right, so again, you're taking a look at a double-stranded piece of DNA. The two strands are complementary to one another. To orient you as far as what you're looking at here, everything here on the far right-hand side, that's what represents the actual gene that encodes for the protein. And so genes are actually really long in comparison to the promoter. So we're only showing you here a portion of the gene. So just pretend that it actually extends off the screen here to the right. The rest of the stuff here in front of the gene, again, that's the promoter region. And so in order to tell where the border is between the promoter and where the actual gene sequence begins, that border, it's given a name. It's considered to be the nucleotide 0. So everything on this side of nucleotide 0 is the gene, and then everything on this side of nucleotide 0 is the promoter that's right in front of the gene. Some terminology that's associated with this is this. So everything on this side of nucleotide 0 is considered to be the area that's downstream. Everything on this side of nucleotide 0 is considered to be upstream, right? So the promoter is upstream of the gene that encodes for the actual protein. One of the things that defines promoters, at least in bacterial cells, is that if you take a look at the -10 region, right? So in other words, if you start at the border 0 and then go back 10 nucleotides, you'll see a region that looks like this. And in every single promoter in all bacterial cells, there's basically the same sequence at this particular portion of the promoter. Same thing here. So if you look at the -35 area, so if you start at 0 and then walk back 35 nucleotides, you'll see that you'll see this particular sequence no matter what promoter it is in bacteria and no matter what type of bacteria it's in, right? So in other words, if you take a look at the promoters of all genes and all bacteria, the -10 area and the -35 area basically look the same in all of those promoters. So that's one of the things that makes a promoter a promoter. It has these sequences at the -10 and the -35 region. Remember that so how about if we sum that up here? So all promoter regions in prokaryotic DNA look the same in the -10 and the -35 region. They have the same sequence there. These sequences in between, we've just kind of drawn in as black lines, as kind of generic black lines, and the reason why we did that is because these are areas within the promoter that actually show some differences among different genes in bacteria. Right? So other regions within the promoter can differ. And so we'll mention the significance of that here in a minute. So again, the promoter, what it does is that it tells the RNA polymerase where to first bind to the DNA so that it can begin transcribing the information in that gene. It turns out that in order for RNA polymerase to bind to the promoter, it actually needs help from another protein. And so in bacteria, the name of that protein is, it's called the sigma factor, right? So that RNA polymerase has to attach to a sigma factor before it's able to bind to the promoter. Right, so again, the sigma factor, it's a protein that binds to RNA polymerase. After it does so, it then allows that RNA polymerase to bind to the promoter. So after RNA polymerase has gotten together with his friend here, the sigma factor, those two guys as buddies then can then bind to the promoter region. After it's bound to that promoter region, and shortly after that RNA polymerase begins to transcribe that gene, that sigma factor actually ends up falling off. So its sole purpose is to help the RNA polymerase bind to that promoter. After it's done that, it ends up falling off shortly afterwards because it's already done its function. And so we mentioned that there can be some differences among different promoters in bacteria, right? So all promoters, again, look the same in this area and this area, but there can be some differences in, this area here, and this area here. So because different bacterial genes can have different promoters, that means that bacteria have to have different types of sigma factors so that their RNA polymerase can then bind to those different promoters. And so here's what I just said in pictures. So if you consider all of the different genes that you find in a cell, each one of those genes has a slightly different promoter sequence in front of it. So these two genes here, gene 1 and 2, happen to have the same type of promoter in front of them. And then these two genes here, 3 and 4, happen to have the same type of promoter in front of them. So we're signifying the slightly different promoter sequences by having them being different colors. So because promoters can differ slightly between genes, that means that in order for an RNA polymerase to bind to that promoter, there has to be the corresponding sigma factor that will help it bind to that promoter. Right? So in order for RNA polymerase to bind to this promoter in front of this gene, it has to buddy up with the appropriate sigma factor that will allow it to recognize that particular promoter. Right? If, on the other hand, that RNA polymerase is going to be able to bind to this promoter, it has to associate with a different type of sigma factor, one that will allow it to recognize that specific promoter. So the idea here is that because different genes can have different promoters, that means that, again, RNA polymerase has to associate with the correct sigma factor in order to allow it to bind to that promoter. Here's why that comes in handy. So it turns out that a cell might not need a gene to be turned on all of the time. So in fact, it might only need it under certain circumstances. And so it turns out that in different environments, a cell will produce a different type of sigma factor that will allow the RNA polymerase then to turn on the corresponding gene that will allow it to best cope with that particular environment. All right, so by having different types of promoters and different types of corresponding sigma factors, it allows a cell to turn on or off genes in different circumstances. Right, so bacteria can make different sigma factors in response to the environment so that it can make the genes that'll best help it live in that particular type of environment. So if we take a step back now and look at the big picture again, we're taking a look at transcription, which is the process of reading a DNA molecule and making messenger RNA out of it. We mentioned that in order to begin transcription, RNA polymerase has to bind to the promoter region, and we've just seen that promoter sequences can differ among different genes, and also RNA polymerase needs a sigma factor in order to bind to that promoter region. What we'll do next is take a look at the terminator region. And remember, the terminator is where the RNA polymerase falls off because it has reached the end of the gene. And so what we'll take a look at specifically is what is it about the terminator region or the terminator sequence that makes RNA polymerase fall off. And so what we'll do is draw that DNA molecule a little bit differently, and so in order to make it easier to explain that. And so we'll draw it so that DNA has already become single-stranded during the process of transcription. These things are color-coded so that this portion here represents the gene. The promoter is green right in front of it, and then the terminator region is red at the end of it. And so remember that what happens during transcription is that the RNA polymerase, assuming that it's bound to a sigma factor, will attach to the promoter. And once it's bound to the promoter, then it goes down and reads that gene sequence and makes the corresponding messenger RNA out of it. when it reaches a terminator, it knows that that's the end of the gene. So the RNA polymerase should fall off now at that point. And so here's what the terminator does in order to make that happen. So it turns out that the sequence that you find within the terminator encodes for a messenger RNA segment that has some self-complementary portions to it. So in other words, if you take a look at this portion of the messenger RNA and this portion right next to it, there are nucleotides that are complementary to one another. So because this portion of the messenger RNA is complementary to this portion of the messenger RNA, something like this can happen. It can fold up upon itself and hydrogen bond to itself. And so whenever you have this structure form, that's called a hairpin loop because it looks like a hairpin. And so whenever you have this structure form, it kind of prevents the RNA polymerase from going any further. So it kind of snags the RNA polymerase so that it cannot, go past that point. And it turns out that this portion here of the messenger RNA, or this portion of the terminator, it's a string of A's, and you know that A's encode for a string of U's. And it turns out that among all of the hydrogen bonds that can form between complementary nucleotides, this is kind of the weakest association. So having this relatively weak bond, A's to U's, coupled together with the fact that this hairpin loop has kind of snagged the RNA polymerase, that's what causes the entire thing to fall off at the terminator. Right? So the terminator encodes for this hairpin loop, and it also causes relatively weak A to U bonds, and that's what causes it to fall off, and that's what causes transcription to end. So to sum that up, here's the answer to this question. How does the terminator cause transcription to end? The terminator encodes for a messenger RNA region that forms a hairpin loop, which is again this structure. That hairpin loop then snags the RNA polymerase so that it basically stops. That messenger RNA then is removed due to the relatively weak A to U bonds that occur after that hairpin loop. We should mention that what we just showed you here, this is just one way that terminators can make transcription end. It turns out that there's other types of terminators that cause transcription to end in a slightly different way, but this is kind of the model that we'll use to explain how terminator sequences can stop transcription. So again, what we've just taken a look at is the process of transcription where DNA is used to make a messenger RNA. We took a look at some of the details of what happens during what happens in the promoter region, and also some of the details as far as what happens in the terminator region. Again, the really big picture, if you keep that in mind, the reason why making a messenger RNA is important is because it goes then to the ribosome in order to tell the cell how to make a protein. So the last thing we'll take a look at is some of the differences when you consider the transcription that happens in prokaryotic cells versus the transcription that happens in eukaryotic cells. And so you might, so one of the differences that we'll point out here is the way that the genes are arranged within the DNA. So if you take a look at how genes are arranged in eukaryotic DNA, it looks something like this, so that each individual gene has its own promoter and has its own terminator. Right? Each gene has its own promoter and terminator. So this means that each one of those genes is transcribed separately from one another in order to make the corresponding messenger RNA, and each one of those messenger RNAs then goes to the ribosome in order to make the corresponding protein. And so to sum up then this arrangement of genes in eukaryotic DNA, again in eukaryotic DNA each gene has its own promoter and own terminator, so each gene is transcribed separately. As a result of that transcription, a type of messenger RNA is produced that's described as monocistronic messenger RNA. So mono means one, so that means that each one of these messenger RNAs that you see here contains the information for making one type of protein. So that's what monocistronic means. It contains info for producing just one type of protein. It turns out that genes are arranged, or they can be arranged, a little bit differently in the DNA of prokaryotic cells. And so, Prokaryotic cells, there are some genes on their DNA that are arranged like this, but there's a different arrangement that you might also find in prokaryotic DNA. And here's what that different arrangement looks like. Turns out that in prokaryotic DNA, a grouping of genes can share the same promoter and the same terminator. So what can happen here is that the same RNA polymerase can transcribe these genes all at the same time. Because there's no terminator between the genes, what ends up being produced is a messenger RNA that it's one long continuous messenger RNA that contains the information that can be used to make several types of proteins at the ribosome. Right? And so then to sum up how prokaryotic DNA gene arrangement is different from that found in eukaryotic DNA, here's what we have. In prokaryotic cells, several genes as a group can be transcribed all together, right? Because there's no terminator in between them like you see in all eukaryotic genes. Whenever you have a gene arrangement like this, a group that share the same promoter and that share the same terminator, what that's called is an operon. One of the things about operons, so because these genes are transcribed all together, they make, again, a messenger RNA that contains information that can be used to make many different types of proteins, right? So in contrast to the monocistronic messenger RNA that all eukaryotic genes can produce, prokaryotic DNA can produce polycistronic messenger RNA. So again, polycistronic means that it's a messenger RNA that contains the information that can be used to make multiple proteins, like you see here. And the last thing we'll mention is this. So one of the advantages to having genes arranged in an operon is that they can all be turned on and turned off at the same time. So typically, if you find genes in an operon, it means that they all produce proteins that work together in order to do something. Right? So for example, in order to break down lactose, you need a set of enzymes that work together in order to break down that lactose. The genes that encode for those enzymes are arranged in an operon kind of like this. So you can turn them all on at the same time, or you can turn them all off at the same time. So if you have a group of enzymes that work together to do something, having one of those enzymes is useless without having the other ones around. And so that's why it kind of makes sense then to make them all together or to make none of them at all. Okay, so again, what we're taking a look at here is what are some of the differences in transcription between prokaryotic cells and eukaryotic cells. One difference is in the way that the genes are arranged on the DNA. Here's another difference in transcription between prokaryotic and eukaryotic cells. So you might remember from cell biology that when DNA in eukaryotic cells is transcribed to produce the corresponding messenger RNA, things happen to that messenger RNA before it can go to the ribosome in order to be translated. So for example, before this can go to the ribosome and be translated, there has to be a string of G's that are attached to one end, and also a string of A's that are attached to the other end. And basically what that does is that it helps the messenger RNA properly attach to the ribosome. Not only that, but things end up being removed from that messenger RNA before it gets translated. So there are regions that end up getting taken out, and then the rest is just put back together. And so after you have that, here's what the messenger RNA then actually looks like when it goes to the ribosome. Remember that these portions that are taken out before traveling to the ribosome, those are called introns, and the regions that stay behind and actually go to the ribosome, those are called exons. So it turns out that this processing of the messenger RNA, so this addition of stuff, this subtraction of stuff, this only happens in eukaryotic cells. So in prokaryotic cells, it turns out that the messenger RNA that's produced right after transcription, that goes as is to the ribosome without any extra stuff being added or taken away. So again, in summary, here's some differences in transcription between prokaryotic and eukaryotic cells. In eukaryotic cells, there is an addition of a G cap and an A tail. And also this removal of introns before translation, that also occurs only in eukaryotic cells. So there are kind of more steps that occur in eukaryotic cells when the information on DNA is used to make RNA.